Well, anyways, it's a pleasure to be here. I should say I spent most of my professional career at Washington University in St. Louis, and I'm a partisan, so I want to make that clear. I got back here last year, and it is a pleasure to be back in Boston, where I was a graduate student. So I work in the nervous system, and particularly on the formation of synapses, and I do that because I think that neuroscience is the most interesting thing in the world. And I actually believe, you may not all agree, I actually believe that most people, if they think about it a little bit, will decide, like I have, that neuroscience is the most interesting thing in the world. Because neuroscience is, as you all know, the study of how our brain works, what the physical basis is, the physical underpinnings of everything we do, of all our behavior, of how we see, of how we perceive, of how we make decisions, of how we execute those decisions. And so our brain really is who we are in an extremely different sense than any other part of us. We're not our kidneys, we're not our liver, we're not our heart, but we really are our brain. And it's a fundamental mystery as to how people, in all their complexity, could in any sense be explained by sort of a lump of soggy cells in the head. And it's not at all clear to anybody whether we can be explained. And so there are many ways in which you can go about trying to answer that, what I would call maybe one of the biggest questions of all humanity is there really a physical basis for who we are, or do we have to go beyond science and beyond the physical? And so, of course, people tackle that through philosophy, they tackle that through psychology, and I've ended up in my life tackling it through biology, with the idea being you sort of start at the beginning, you start at the lowest level, and figure out what you can and build up, and at the end there may be something left that's unexplained, and that is something that's going to be beyond science. It's going to be maybe our mind in a way that can't be explained by our brain. And I don't believe for a minute that I'm going to get there in my lifetime, but I really do think I'm part of a process that will eventually get there and answer that big question. Is there something about us that can't be explained by physical laws? Now, separately from that, and maybe a more practical application, the study of neuroscience is now of tremendous and growing clinical importance. And that, of course, motivates many neuroscientists, probably me less than others. It will motivate many of your students. And essentially, the brain diseases, I would say at this point, are the most mysterious and the most costly of any diseases we have. The neurological diseases and the psychiatric diseases, the line between them blurring more and more as we go on, really cause more morbidity, more loss of work, more loss of happiness, more loss of time than things like cancer or heart disease. And even in my short scientific lifetime, what neuroscientists know has contributed tremendously to our approach to those diseases. So when I was a student, neurology was a diagnostic science. That is, people would go into the clinic and they'd learn how to diagnose extremely accurately where the lesion was if you had had, let's say, a stroke or had multiple sclerosis. They could tell you that probably where your brain had gone awry was right here, right here, right here, and then there's just nothing whatsoever that anybody could do about it. And in the past 20 or 30 years, based on what we've learned about how neurons work and how they fail to work and how we can change their activities, there are really treatments that, if not fully in the clinic, are definitely in clinical trials. Pills for multiple sclerosis, pills for ALS, pills for Alzheimer's disease. And it'd be a lie to say that people are getting better yet, but it's pretty clear that help is on its way. As I say, things are in advanced clinical trials. There are already things in the clinic. For the psychiatric diseases, which may be neurological diseases as well, we just sort of call them psychiatric because we can't figure out what part of the brain is broken. There was already treatment a long time ago. You all know there were psychoactive drugs. Probably the first generation were things like Thorazine for schizophrenia, Librium for anxiety, and so forth. But they weren't terribly effective. And new ones are more effective. Risperidol is a better treatment for schizophrenics than Thorazine was, and Prozac is a better treatment for depressives than the MAO inhibitors were, and so on and so on and so on. There are treatments for OCD. And that's all good, but they remain incompletely effective. And I would say that the reason they're not effective is that unlike the neurological diseases, we still have no idea whatsoever what goes wrong in those psychiatric diseases. And so the treatments are all trial and error. There's not yet a rational way to devise good treatments. And so I'd say one of the main sort of hopes for the future for neuroscience is to really explain the psychiatric diseases so that they can be put more fully into a medical model. So that is sort of what got me into the field, what's made me spend my life in it. Now, what I want to do today is actually not talk about any of that. That was more or less by way of introduction, but do two things. The first is give you what is probably going to be a review for most of you, just a few basic facts about how the nervous system works, how neurons function. And again, I don't mean to be condescending. I know many of you teach this, but having done this before, I realize people are at different stages, and since you'll be getting a lot of more advanced and more sophisticated talks than mine over the coming weeks, it's probably worth being on the same page. And then I want to take the rest of the time, or most of the rest of the time, showing you some pretty pictures. The point being to give you a sense of how nerve cells really look. And the sort of subtext of that is, while I'm showing you how they look, to give you a sense of what are some of the new ways that people have devised recently to find out how they look and to watch neurons, and particularly to watch neurons in living animals. And that's really one of the revolutions in today's neuroscience, that we don't have to kill, fix, and stain an animal to see nerve cells. We can, at least in some cases, watch the neuron in life, and as you can imagine, once you can watch it in life, you can watch it over time, and you can see how it changes. You can see what happens to a neuron when you learn something, what happens to a neuron when you get old, and so forth. And the we, in this case, is mice, but you know, the hope is that what we learn there will be applicable to people. And then at the very end, sort of along the same theme, I want to say a little bit about how synapses work, the connections among nerve cells, and how we look at them, and there we're getting closer to what my own research is about. So let me just give you the very briefest of reviews. The fundamental unit of the nervous system is the neuron. It's a cell, much like any other cells. It has a cell body. It has a nucleus where its DNA is, but it's different than other cells in that it has extremely long processes. And those processes are of two types. Many neurons, not all, but many, have a single very long process called an axon that comes out of its cell body, which is called a soma. And again, many but not all neurons have a larger number of shorter, fatter processes that are called dendrites. And basically, the way neurons work is to conduct electrical signals along their processes. And in general, they start from the dendrite, they pass through the dendrite to the cell body, they keep right on going out the axon, and they come to the other end. And the reason they go in that direction is less because there's an intrinsic directionality in the neuron, but because neurons are connected to each other by connections called synapses, which I'm going to be telling you a lot about. And the general pattern of connectivity in the brain is that the axon of one neuron makes a synapse on the dendrite of another neuron. And so what that means is that an electrical impulse passing along an axon will get to a synapse. That will then lead to the excitation of the dendrite, which will initiate another impulse, which then passes on down through the cell body or soma to the axon, eventually perhaps to another dendrite, and on and on and on. And the way the brain really works is by wiring together huge numbers, and huge numbers means millions or billions of neurons, into circuits such that every neuron may receive tens or hundreds or thousands, or even in some cases tens of thousands of different synapses to different neurons from different neurons, and its axon then will branch and make tens or hundreds of connections onto different neurons. Now, this arrangement actually has a couple of interesting consequences that I'll just mention very briefly for those of you who sort of maybe more familiar with general cell biology. So one is that the cell body of the neuron, like any other cell in the body, has its nucleus. The nucleus has DNA. The DNA is transcribed to make RNA. The RNA is translated to make protein. But all of that is going on, or at least 99.9% of it is going on in the cell body. But that protein that's made has got to feed these enormously long processes. And it's not a difficulty that most cells have to face. A kidney cell is sort of just like a little sphere. It's like this without the processes. And so there are phenomena that go on in neurons called transport, such as axonal transport, that take the proteins made by the normal transcription and translation machinery in the cell body and then ship it down to axons or ship it out to dendrites. And as you can imagine, one of the sort of interesting problems that comes up is how do you get the right proteins to go to the right place? How do you get those that need to go to the axon to be there, those that need to go all the way to the synapse to get all the way there, and those that should have stopped to stop, and so on and so forth. So although in most ways neurons are sort of like other cells except funny shaped, they do have this one extra set of cell biological phenomena going on in them. And then they have a second extra set of cell biological phenomena going on in them, which pertains to the fact that what they are doing, much more than any other cell, is transmitting information. And so the basic way in which neurons transmit information is as follows. They do it by electricity. I'm sure you all know that. And the way they generate electricity is by having a gradient of ion concentrations across their membrane. So if we imagine just a little bit of membrane here, I'm going to give you an explanation that is, I would say, not strictly true, but it's true enough to give you a sense of it, and we can talk about the particulars later. So the axon membrane, which I'm now blowing up many-fold, is made of lipid, of course, like all membranes, and as such, it's impermeable to ions, almost. And the way it works is that there's a very high concentration of sodium ions on the outside and a fairly low concentration of sodium ions on the inside, and exactly the opposite for potassium ions. So things are pretty well in balance. But it turns out that there are a few little holes called channels in the membrane that are selectively permeable to potassium. So although everything's in balance, when these potassium channels open up, a little bit of potassium can flow out, and that leaves the inside of the cell with slightly less positivity than it had before, and the outside with a bit more positivity. And so what that means is the membrane essentially becomes like a battery where there is a net negativity inside and a net positivity outside. Okay? Now, when an impulse flows along a neuron, the way it accomplishes that feat is by making use of not these potassium channels, but of sodium channels. And these are proteins whose structure is now extremely well understood. In fact, they're X-ray crystallographic structures of them. A guy named Rod McKinnon got the Nobel Prize about 3 years ago for coming up with the X-ray structure of the first potassium channel. So a sodium channel opens up. That now lets a little bit of sodium come in, and that basically gets rid of the membrane potential. So the cell goes from being inside negative to neutral, and it even overshoots. So if you were recording with an electrical device, you would start with the cell with a negative potential, and it's on the order of 1/10 of a volt, or 100 millivolts inside negative. Then the sodium channel opens, and the membrane potential gets high. And then the potassium channel hyper opens, and that lets in more potassium, which is now counteracting the sodium, right? So it lets out more potassium to counteract the sodium coming in. Then that brings the membrane potential down, and then everything closes and you start all over again. And so the way an electrical impulse flows along an axon can be described in the same way electricity flows along a wire, but in fact the physical basis is quite different. Instead of it being electrons bumping into each other along a wire, as you remember from physics, it's actually these sodium and potassium channels opening, closing, opening, closing, opening, closing, opening, closing, leading to this sort of wave of depolarization and repolarization moving along the axon. Now parenthetically, of course, you'd realize that this couldn't go on for very long. After you, let's say, ran around the block one time, all your sodium would have come in, all your potassium would have gone out, and you'd be dead. And the fact that that doesn't happen lets you know that there must be some way to recharge the battery. And that way actually is a little enzyme called a pump, which when you're not doing action potentials, takes care of pushing the potassium back in and pushing the sodium back out to reset the gradient. That's a detail we don't need to deal with because it happens over a very slow time scale, so it doesn't really affect the transmission of information. Okay? So that's what I wanted to say about how neurons look and about how they conduct electricity. The last part of the introduction is about what happens at the synapse in between neurons. Because that, and it's a point I want to come back to, is where the information processing really happens. To a first approximation, the neurons are sort of like wires, whereas the synapses are more like the transistors and the diodes and all the machinery that does the really fancy stuff. So here's how a synapse works. And again, let's blow this up a little bit. So let's say this is the part of the axon that's presynaptic, and this is the part of the dendrite that's postsynaptic. So an action potential is coming along, a wave of polarization, or in this case depolarization, and now it comes up to the nerve terminal. Inside the nerve terminal are little membrane-bound packets called vesicles that contain within them a chemical called a transmitter. When the electricity comes along, what it does is now open yet another channel. This is now a calcium channel. And the way it does that is pretty much the same way it opens the sodium and potassium channels that I already told you about, except this is a different protein, and instead of being selectively permeable to potassium or selectively permeable to sodium, it's selectively permeable to calcium. Why that's important is that calcium, when it comes in, serves as a cofactor to cause these vesicles to fuse with the membrane. And when they do that, their transmitter diffuses out into the extracellular space, and particularly the space in between the axon and the dendrite, which is called a synaptic cleft. Now, this is a very complicated system, and it's something that's worth thinking about, why nature selected such a very complicated way to make a synapse transmit information or pass information when you might have thought it could just sort of solder the wires together the way you or I might have done. And we can come back to that later on. At any rate, the transmitter comes out, calcium's come in, it's caused vesicle fusion. Believe it or not, that can happen in about 2 milliseconds. The neurotransmitter diffuses out. The distance is so low that it can diffuse across the synaptic cleft in a few microseconds. And then the transmitter interacts with a receptor protein, and I'll show you a picture one later on. And the interaction of the transmitter with the receptor opens, believe it or not, yet another channel which now lets ions pass into or out of the cell and start the depolarization and the action potential process again. So the whole thing is working by ions that are kept apart from each other by this membrane, which is impermeable to ions, but is made selectively permeable by proteins called channels that are selective to one ion or the other, and that can be opened very specifically by one thing or another, by depolarization itself, by neurotransmitter, and there are many other sorts of things. So the upshot then is an action potential comes along, comes to the nerve terminal, lets in calcium, causes a vesicle to fuse with the membrane, neurotransmitter comes out, diffuses, opens a channel, lets in more current, eventually leads to the generation of a new action potential, moves along the axon, connects up to another synapse, and so on. And then all of these millions of neurons are working together in very specific and particular ways as circuits. And in the end, we think that you behave. You make decisions, you have emotions, and what have you. And going back to what I said at the beginning, the overarching question in the field is, can this explain why you're happy some of the time and you're not happy some of the time, and you're really clever some of the time, and you do really stupid things some of the time? And that, I would say, remains an open question.
Could I just ask you on the neurotransmitters, you're saying the postsynaptic neuron, the calcium ions are at the postsynaptic neuron, in the postsynaptic neuron?
The calcium ions are in your blood. They're all over everywhere on the outside.
Okay, so because I thought you were showing them coming out.
I did. I'm just not a very good blackboard artist. What I meant is I was just sort of drawing an arrow to where the calcium comes in. I didn't mean this. I wasn't trying to say it was inside the cell.
Now, with some of the medication, from what I understand, it allows some of these neurotransmitters to remain for a longer duration at the synapse. Are you going to talk a little bit about that?
I wasn't going to talk.
How does that work?
Yeah. So how does that work? Well, you know, one sort of general statement is that most of the psychoactive drugs seem to work at synapses one way or another. And one of the ones you may be thinking about would be SSRIs, serotonin reuptake inhibitors, of which Prozac, Zoloft, the modern antidepressants are examples. So what they do is prevent the uptake of serotonin so that the serotonin is here longer, lasts longer, and presumably activates more receptors. So one way of thinking about what an antidepressant does is that it compensates for some kind of endogenous deficiency in serotonin. You don't have quite enough, and so you make what you have last longer. Now, it's turned out that's way more complicated than that, but that certainly is a part of the story. And I should say, you know, the other drugs I mentioned before, the antipsychotics, other antidepressants also work at synapses, as do recreational drugs that you may see from time to time. Cocaine does, marijuana does, heroin does, morphine does. They all have actions that we're beginning to understand fairly well at the level of particular synapses. They interact with different neurotransmitters. That's what gives them their specificity. This is how people thought neurons looked for the longest time. This is a section, actually, of a chicken brain from work done in my lab. And it was prepared in the conventional way, in which you kill the animal, you fix the brain in something like formaldehyde, formalin, then you cut thin sections of it, and then you stain those sections with a blue dye that sticks to cells. And this is what you see. And you can learn quite a bit about it. This would be the top near the outside. This would be the inside where the ventricle is. And it's probably quite plain to see that the neurons aren't randomly arranged. They're sort of arranged in laminae, in layers. So there's a lot of neurons here, fairly few here, more here, and so forth. And by looking at sections like this, people learned a tremendous amount about the organization of neurons. But obviously there are limitations, many of them. One of the ones that maybe would strike you is that essentially you've got all the neurons labeled, and so you're in danger of just having a big blue mess in which you can see that there are lots of neurons there, but because they're all packed together, you can't see very much detail about any individual one. And that state of affairs held back the progress of neuroscience for a long time, until the beginning of the last century, 1900 or so, when this guy, Santiago Ramon y Cajal, found a way around it. So I'm showing you his picture because he lived and worked in about 1880 to 1920 in Spain, and he was hands down the greatest neurobiologist who ever lived. And so it's worth remembering his name, because there probably aren't that many fields of endeavor in which you can point to a single person and say this was really the greatest by a wide margin. And he did many, many things, but one of the things he did was to apply a stain that actually an Italian man named Golgi had devised, that for reasons that we still don't understand, stained only a very small set of neurons in the brain, more or less at random, remains a mystery, but it stained that random subset in exquisite detail from the tips of their dendrites on the incoming side to the ends of their axons on the outgoing side. And he used the Golgi stain to really map out for the first time and in a systematic way what neurons look like in the brain. And these are just two pictures that I took from truly thousands that he made. This is one of the most complicated neurons. It's called a Purkinje cell. It's in the cerebellum. It seems to be involved in motor coordination. And it's one that has tens of thousands of synapses made on its dendrites. Be one of the more complicated ones, but has just a single axon going out. That axon branches, and this is just the beginning of it. It'll end up making many branches and contacting many synapses on the way out. Here's a picture from the cerebral cortex in which he sort of graphically superimposed many neurons from different preparations as he began to build up a picture of how neurons connected with each other. In fact, he was also the first person to see that synapses existed. He couldn't see them in detail because he didn't have high enough resolution, but he found that neurons ended in axons that were in apposition to other cells, and contrary to the prevailing view, which was that all neurons were essentially connected together in a syncytium, meaning a continuum, he was the guy who really showed that there are discrete specific circuits connected by discrete patterns of synapses. So this work went on for a long time, and it helped people map out some circuits. It helped Cajal learn a lot about development. I'll say a few things about that later. And he was even able to infer sort of what some of the circuits were just by looking, even without being able to record their electrical activities. And it's sort of magic. I would say to this day, nobody knows how he was able to figure that out, but he did. He wrote down what he thought, and in almost every case, when people have, a century later, been able to record electrical activities individually from cells, he turned out to be right. So over the 20th century, one of the things that people did, especially second half of the 20th century, was to devise methods to label individual cells, but not at random, rather to target particular ones. And this is just one of many, many methods. It uses antibodies, which you may know you can make to be very specific for particular components of neurons, and then you can use those antibodies on these sections to stain the particular neurons that contain the particular protein, and I won't go into the staining method, but that allowed people eventually to find out something about the molecular profile of individual neuronal types and find out the connections of, let's say, for example, go back to the Prozac example of those particular neurons that either made or responded to serotonin. It's tremendously useful. But it still was limited in many ways, one of them being that you can really only do this on fixed tissue. And so you can make inferences about what happened in live animals, but you can't really know for sure because the animal's not live anymore. And so I would say one of the biggest advances in neuroscience and maybe in biology generally came about 10 to 15 years ago thanks to this animal, the jellyfish. And the jellyfish, this jellyfish, and actually a lot of jellyfishes, you may know fluoresce. You may have seen this down at Cape Cod. If you go to the Cape Cod, to the Cape Cod seashore on a moonlit night, you can sometimes see the water glowing, and it's glowing because there's lots of jellyfish in it. And you can sort of see that fluorescence if you take a jellyfish and illuminate it with the black light. So you illuminate it with ultraviolet light and then see what kind of light comes back. And this is an example of a picture of this jellyfish illuminated not with white light, but rather with ultraviolet light. And this particular jellyfish seems to have around its edge, around its tentacles, a compound of some sort that fluoresces green when it's illuminated with ultraviolet light. So a guy named Shimomura, actually 20, 30 years ago, decided to find out why this happened. And he did it by a biochemical approach of taking jellyfish and essentially grinding them up and beginning to purify different proteins from them until he finally purified a protein that had the property as a pure protein that the jellyfish did as a whole organism, that it would fluoresce green when you shone ultraviolet light on it. And that protein he called green fluorescent protein, not very imaginative, and it's called GFP today. This is a picture of its X-ray crystallographic structure, and it actually is, even the structure of it is quite beautiful. And I don't have time to go into why people think this structure leads to something fluorescent. In fact, I'm not even sure I understand it. But once this had been done, what people realized was that they could implant this green fluorescent protein into other cells and make them glow. They could do to other organisms, other cells, what the jellyfish had done for itself. And so, for particular relevance to neuroscience, what people began to do was to make transgenic mice in which the green fluorescent protein, or in this case a yellow fluorescent protein, I'll come to that in a minute, was put into the mice so that yellow fluorescent protein itself would be made in neurons. And very briefly, you may know there are methods to generate transgenic mice in which you micro-inject DNA into the nucleus of a fertilized egg, and that DNA will actually integrate into the chromosome, and it will become a gene like any other mouse gene. So as that mouse develops, it has the green fluorescent protein gene in this case in every one of its cells, and all of its babies will have the green fluorescent protein in all of their cells, so you don't have to do it over every time. And by hooking this protein up to the proper regulatory elements, you can make it be such that even though every cell has the yellow fluorescent protein gene, only particular cells will make the yellow fluorescent protein. And again, that's just the way all our cells work anyways. You know that all your cells have the myosin gene, but only your muscle cells make myosin. All of your cells have the globin gene, but only your erythrocytes make hemoglobin, and so on and so forth. And so this is essentially what you'd call a neuron-specific promoter, driving YFP. And when people began to make those mice, what they could then do was look in the brains of those mice and now see in exquisite detail those neurons that express the yellow fluorescent protein. And here's an example in a mouse brain of a particular transgenic mouse in which the protein was expressed selectively in a particular layer of the cerebral cortex and in the axons of those cells. These cells actually include your upper motor neurons, whose axons are going to go down and innervate your lower motor neurons in your spinal cord, and here you can see their axons heading down. Now what was neat about it was that this fluorescent protein allowed very detailed filling and visualization of the neurons. And so this is just a higher power of the mouse that you saw before, where you can now see these individual neurons. They're long dendrites. Remember, the dendrites branch up there. There's so many axons that you can't make out individual ones. But there were some mice in which it could be arranged that only a few neurons would be labeled. And in those, you can, in sort of the same way Cajal did, come to see details of the structure of the cell body, the dendrites, the axons, even these little spines, which turn out to be the postsynaptic sites on the dendrites where the synapse is formed that I was drawing for you a little while ago. Now, I'm going to come back again and talk about neurons, but the main point that again I'll come back and show you in a slightly different context is that first of all it's very pretty, and that was very nice because this is in living color, whereas Cajal had to work in black and white. But the real advantage, and again, I've sort of put the example a few minutes down the line, is that you can now illuminate brain tissue with an ultraviolet light in life and see the neurons in life. Whereas for previous staining with antibodies or with the Golgi method or with the blue stain, it only worked when you fixed the tissue and broke it open. And so that's what paved the way for seeing how these neurons look in living animals and then seeing how they change over time. Yep.
Is that particular neural gene always turned on in neural tissue always, or is it turned on and off?
Yeah, so that is sort of under the control of which particular gene you take the regulatory elements from. So this one that I'm showing you, which we've used a lot in our lab, turns on around the time of birth and then stays on forever.
In neural tissue.
In neural tissue. But there are certainly other ways to do it, so it comes on early, goes off late, is on forever, and craftier methods are now available to turn it on at any time in adult life just when you want it on. And we can talk about that later. Okay, so before I come back to sort of looking at these things in living animals, let me say a little bit about synapses, which is what I work on. And then in the context of synapses, I'll tell you a little bit about live imaging, which is really the big deal about GFP or YFP. So the synapses that we study are neuromuscular junctions. And those are the synapses that motor neurons make on muscle fibers. They are, I would have to confess, a lot less interesting than synapses in the brain because they pretty clearly don't learn very much. They don't seem to be very involved in emotional behavior. They're not involved in complicated circuits. But they have a couple of technical advantages that made them a good first synapse to look at for many developmental issues and also for the live imaging, which is kind of the theme of what I want to talk about today. So let me just introduce you to them. So as I said, neuromuscular junctions are the synapses in your muscle. The cell bodies that make those synapses are in your spinal cord or brainstem. They have extremely long axons that extend out through the peripheral nerves, all the way out to individual muscles. And then when they get to those muscles, they branch and innervate individual muscle fibers, the long cylindrical cells that make up muscle. The long cylindrical muscle fibers are really the contractile elements in a muscle. And all the axons in most of the muscles enter through the middle, and then they form branches, and then those branches terminate on individual muscle fibers in a little sort of claw-shaped or pretzel-shaped structure that I'm going to show you a real picture of in a few minutes. Now, if you look in the electron microscope at sort of a section through one of these muscle fibers, what you see is the structure of the synapse, now drawn a bit better than I drew it on the board before, but with roughly the same elements. So here is the axon that's come all the way out from the spinal cord. Here it's sort of made bulbous, where it contacts the muscle fiber. Here are the synaptic vesicles that contain the neurotransmitter. Here are the active zones or release sites that have those calcium channels associated with them, so that when an action potential comes here, the calcium enters right here, and this is where the vesicles fuse. The transmitter goes across the synaptic cleft. There's material in the middle, but it's very porous, so it doesn't get in the way of the neurotransmitter. And the neurotransmitter, in this case acetylcholine, then gets to the postsynaptic membrane, where it interacts with the acetylcholine receptors that then open, let through current. And in this case, the current initiates an action potential that causes contraction of the actin and myosin. If it were a neuron, the action potential would just flow along until it got to another synapse. Here it's sort of the dead end. It's the final point of the nervous system. And the effect of the action potential is to cause muscle contraction. And here's just a real electron micrograph showing you what one of these synapses actually looks like. Again, the vesicles, the release site, the synaptic cleft, the neurotransmitters. This is a muscle from a mouse that expresses the yellow fluorescent protein in its neurons. It's actually a mouse very much like the one whose brain I showed you before. And what you're seeing here is sort of a big, thin, flat muscle in the neck of a mouse. The muscle fibers are running vertically. You can't say, they're not really stained, but if you look hard, you can sort of see a striation. That's the individual muscle fibers. The axons are all entering through the muscle nerve, as I showed you in the sketch. And then individual axons branch, leave the nerve, and eventually end on individual muscle fibers. And in this particular case, what we've done is to stain the acetylcholine receptors with a red stain that binds to them in living animals, because the receptors stick out, and so you can get to them with this red stain. So what you're seeing here is the YFP labeled axons, the red postsynaptic receptors, and the overlap where the axon ends and the receptor is the perfect apposition of the pre- and postsynaptic structures that characterizes the synapse. Now you can look at one of these areas at higher magnification and see it a little bit more clearly. So here now we're looking at the individual axons in a motor nerve, and we can see them sort of branching many times and forming their synapses. Each of these synapses is on a muscle fiber. We just haven't stained the muscle fiber. And the way the arrangement is in the skeletal muscles of mammals is that every axon branches to innervate maybe 20, 30, 40 muscle fibers, but every muscle fiber receives just a single synapse. So it's a bit different, much less complicated than the brain. Now, a good thing about this yellow fluorescent protein is that it really fills the nerve very, very completely. And so it's possible to look at very high magnification and see in a lot of detail just what the structure is. And so in this case, we're now just looking at a couple of axons, a couple of neuromuscular junctions. Here we've labeled the contractile apparatus blue so you can see the muscle fiber. And again, you can see the axon. It's sort of pretzel-shaped claw of endings. It's lying atop the red receptors. The apposition of the green nerve to the red receptors is so perfect that you can just barely see a few little shreds of red sticking out where the green isn't. Now, one main advantage, I should say, of looking at the neuromuscular junction, I probably should have said this first, is that it's extremely big. So it has one advantage in terms of this live imaging, that it's out in the periphery, and therefore surgically access to it is relatively easy, so we can look at these animals in life, as I'll show you in a second. Another advantage is that it's a huge synapse. So these are synapses in the brain, 3 of them shown at the same magnification as the neuromuscular junction. And in both the cases, the presynaptic structure is green, the postsynaptic structure is red, and their overlap is yellow. And so the point being that if you want to see whether a synapse is present in the brain, you can do that. But if you want to see how it, say, grows or remodels or changes with experience, or how stable it is, that's really quite difficult. And so that's the second reason, besides the accessibility, that we began by trying to do live imaging of neuromuscular junctions. And a lot of the experiments that we've done have involved just watching neuromuscular junctions. So I, by background, am a molecular biologist. I've been very interested in finding out what are the molecules that are involved in making a synapse form or making a change. But in this project, we've just gone back to have a look and see what's there so that we really know what's going on before we try to find an underlying mechanism. So the methods for imaging were actually devised by my colleague, Jeff Lichtman, whose lab is next to mine here, and whose lab was next to mine in St. Louis as well. And a long time ago, he devised methods to actually make an incision in the neck of a deeply anesthetized and respirated mouse and expose a superficial muscle. And given that surgical ability, he can then take a microscope and position an objective right down on the muscle and look at it. And here's a picture in life of the incision. This is now a dead mouse, and the incision is way bigger than it would ever be in an experiment. But you can actually see the muscle. The nerve would be here. You probably can't see it from where you're sitting. And you can then actually look in now, turn off the white light, turn on the UV light, and in these XFP mice or YFP mice, see the neuromuscular junctions. And you can then, say, find a neuromuscular junction, take a picture of it, and that's what was done here. Then take that animal, put a few stitches in the neck, because the incision isn't as big as the one I showed you, revive the animal, return it to its cage. It'll run around, be perfectly happy, and then whenever you want, a day later, a week later, a month later, a year later, you can take it out, and in most cases find the very same neuromuscular junction and see whether it's changed. And one of the discoveries that Jeff made was that actually for neuromuscular junctions, they don't change terribly much. Here you're looking at a synapse that was imaged on the 40th postnatal day and again on the 47th postnatal day. And you can pretty much see that it's the same synapse, the same nerve. You can hopefully see that this branch with this little thing coming around is this branch with this little thing coming around. These two dots are these two dots. This arc is this arc. And most of the changes probably just have to do with a little bit of mechanical tension from one view to the next. But that stability has been sort of interesting because it's a great baseline, and then you can find out how it is that growth or aging or activity or inactivity or various perturbations might make the synapse change over time. In the very end, what I'm going to tell you is that synapses in the brain, which are much smarter, learning all the time, are actually not that stable. They really do move around, and if you're listening carefully, there are probably synapses at this very moment moving in all of your brains. So this is imaging done sort of day-to-day, week-to-week, month-to-month time scale. But it's also possible to image developmental events. And here's an example of that. So what was done in this case is the following. This is an adult mouse, and a graduate student in the lab, Quinn Nguyen, did the following experiment. She opened the mouse and cut the nerve. And you may know that if you cut the nerve, the axon is separated from the cell body. You remember that's where all the protein synthesis is going on. And that axon will degenerate and go away. Now, if the axon that's been cut is in your brain or spinal cord, that's pretty much the end of the story. Regeneration is extremely poor in the central nervous system. That's why spinal cord injuries, for example, are so devastating. In the peripheral nervous system, however, regeneration is not so bad. And so people are very interested in looking at regeneration in the peripheral nervous system to see if they can find out what are the critical differences between the peripheral and the central nervous system so that they can improve regenerative ability in the brain and spinal cord. And that's what this experiment was about. Yes? It's not just slow. I mean, it'd be an exaggeration to say it's non-existent, but it's more than slow. I mean, it mostly doesn't happen. Yeah. In other words, it's not the case that if you wait long enough, it'll finally happen. It's that very little happens. What happens pretty fast, just not much. Okay, so in this case, what Quinn did was the following. She cut the nerve. She waited for it to degenerate. She knew from previous experiments when the axons would have then grown back into the muscle. She reopened the animal at a time when she knew it was the right time. And she looked for one of these neuromuscular junctions with now the receptors labeled, at which an axon, to which an axon had almost returned. And then in this case, she turned on the movie camera and watched what happened. And here, over a period, there's a time stamp on it of about 4.5 hours. This axon came back and reinnervated this neuromuscular junction. It followed the original pathway. That alone is interesting. It turned where the original postsynaptic membrane turned. It branched where it had branched. And when she went home for dinner at 4 hours and 39 minutes, about half the synapse had been reinnervated. She came back the next morning and the other half had been reinnervated. I just don't have that picture here. So she actually saw, probably for the first time ever in a mammal, a synapse forming. And obviously it's now quite interesting to use this system to find out what is it that improves matters or makes matters worse. Now, a lot of the work in my lab involves looking at initial synapse formation in embryos rather than reinnervation following nerve injury. And that still is almost impossible to do in mice, not because YFP isn't good, but because the mother mouse is opaque rather than transparent. And so one way to get around that is to do the same sort of experiment in little transparent fish called zebrafish, which are quite wonderful, easy to keep, easy to label with YFP. It might be something you might want to think about using in your classrooms. And these are fish in which the motor neurons now are also labeled with YFP, But because the fish is transparent, Heather Flanagan-Steet, who was a student in my lab, was able to make a movie of the initial formation of the synapse, the growth cone, the growth part of the axon coming out and beginning to make these synapses. And for reasons of time, I'm not going to tell you exactly why we wanted to do this or what the conclusions are, but just kind of make the technical point that that's now possible. So let me finish just in the last, I think, 2 or 3 minutes, giving you a sense of where this technology is going. And I'm putting this picture here to sort of illustrate a problem with it. And the problem is as follows. If you want to look at a synapse, as I think I've showed you, this is a wonderful technology. But if you want to work out wiring diagrams, it's not very good. And the reason is illustrated here. Even in something as simple as a muscle, the wiring diagram is too complicated. There are all these axons, and they're making all these synapses, and you just can't follow them very well. They're all tangled up like spaghetti, and so you can see that there's an axon here and an axon here, but if I asked you to tell me whether it's the same axon that makes these 2 neuromuscular junctions or these 2 neuromuscular junctions, obviously you just couldn't do it. And so what one needs to get around that is a way to label just a few axons. And there are now transgenic methods to do that. And this is showing you side by side a picture from a muscle in which all the motor axons are labeled with YFP, or just one axon is labeled by YFP. And I think the point is clear. On this side, this muscle is fully innervated. It's just most of the axons are invisible under black light. Only the one is visible. And now, if I ask whether this synapse and this synapse come from the same axon, the answer is trivial. And so using these single axon mice, it's possible to actually reconstruct the wiring diagram of a single motor neuron. And that's what was done here by a student in the lab called J.D. Wiley, who traced and reconstructed the pathway of this motor neuron and found all of the synapses that this particular motor neuron makes on muscle fibers in this muscle. Again, all the other red spots are muscle fibers that have synapses on them. You're just not seeing those synapses. And so now you can ask a lot of other questions. Do all the synapses made by a motor axon look alike? If there's a variety of synaptic types, a variety of synaptic responses, or a variety of, let's say, responses to stress, is that mediated at the neuronal level? Do all the synapses made by a single neuron behave the same? Or does any neuron make as wide a variety of synaptic types as is present in the muscle as a whole? Simple questions that people have asked for ages, but just didn't technically have the means to answer. Now, there's still a problem here, and that is that even though you can now see a single axon, as I've already said a couple times, you can't see the other axons. They're invisible. And so a way around that came from work in many different laboratories, which showed that it's possible to actually make variants of the green fluorescent protein that fluoresce in different colors. And in fact, right at the start, I was showing you yellow fluorescent protein, which already was a variant. But using this, it's possible to make transgenic mice that now have yellow fluorescent or green fluorescent or cyan fluorescent or red fluorescent neuromuscular junctions. And then what you could do is say, for example, take a mouse that is so constructed as to have, let's say, a single blue axon in a muscle, and mate it to a mouse that has all yellow fluorescent axons in its muscle, and by the laws of genetics, in 1/4 of the babies, there will be both transgenes, and you get something like this. You get a muscle in which all the axons are yellow and just one is blue. And using that, then, you can really, first of all, look at circuitry and compare your favorite neuron to all the other neurons. Then you can go on and do something else. You can really begin to look at individual synapses and find something that I guess people knew but couldn't study, and that's shown here. So remember I told you every neuromuscular junction is innervated by a single axon. Okay, so every axon branches to innervate 20 to 40 neuromuscular junctions. Every muscle fiber gets a single neuromuscular junction, but that's obviously not the case here. Here's a single neuromuscular junction that's innervated by 2 different axons. And people actually knew that, and they knew that that's the case in early postnatal life. And then there's a process of synapse elimination whereby these axons seem to compete so that one is lost and the other is retained. Sometimes there are 5, and then 4 are lost and 1 is retained. And that goes on in the brain as well. It's very well documented, and it probably has something to do with the critical period. This is probably a big part of the explanation of why our brains are extremely plastic in early postnatal life, because we have a lot of connections amongst which we can select. And at some point, most of the connections are gone, and those that remain are more stable than any of them were before. You can't do that selection anymore. In the brain, it's called the critical period, and we know it in many ways. We know that little kids can learn a lot more and learn a lot of different things young than they can old. Language learning is probably the simplest example. There is a time when you can learn a new language and you can get a great vocabulary, but you'll never learn it without an accent, no matter how long you live. And that may have something to do with sort of the fact that you have many potential connections early on, and in the end things stabilize. At any rate, here out in the neuromuscular junction, what you can do now is actually watch that competition happen. And so here's a series of pictures from a student in Jeff Lichtman's lab named Mark Walsh, who found a neuromuscular junction in very early postnatal life that was innervated by one yellow and one blue axon. And he took a picture of it, and then he came back the next day, and of course what looked to be the case was that the blue axon was kind of pushing out the yellow axon. What happened was that that wasn't the case at all. In the intervening day, the yellow axon had made a startling comeback and was beginning to push out the blue axon. When we came back the day after that, the yellow axon was continuing. Next day, the blue axon had pretty much given up the ghost, and the next day it was really gone altogether. And so what he was able to see, of course, was synaptic competition happening in living animals. And that, again, I mean, I don't need to belabor the point. There are many, many sorts of questions you can now ask about that. And so the future is, I think, to use these methods now to approach questions in the brain which we always wanted to study, but which we just didn't have technically the tools to study. And I've already shown you that this YFP works in the brain. And indeed, neurons, in this case sensory ganglia, but also in the brain, can be labeled with different colors of fluorescent protein, and you can do those same tricks of mating one color to another color, and then get in the brain sets of neurons that are, in this case, yellow or blue, or here's one that's yellow plus blue. So you're already sort of getting a third color. And one of the main activities in our lab now is to get more and more colors and various tricks that we can talk about later. And here's a picture from the brain of one that now has 5 or 6 different colors. And so the deal would be now that as people develop methods to actually optically look in the brains by thinning the skull and using the same sort of optics that I told you about, and several labs have done that, you can actually go in and begin to see synapses form or change or rearrange in life. And then the trick is, and there are already people doing this, not in my lab, but in other labs, you can teach a mouse something and see whether synapses form or are lost or get bigger or get smaller. And so really begin to get to where I started you off an hour ago, really looking to see what's the physical underpinnings of mental activities. I don't know if you'd want to say a mouse is all that smart, but I think it's pretty cool that we can do this by now.
Probably a foolish question. How do you get back to that same?
Yeah, no, not foolish. It's a huge problem. But basically they're landmarks, and some of the simplest landmarks are capillary blood vessels on the brain. And so you sort of take a low power picture at your first view and you see a map of capillaries, and that's a big help. And then when you come back, you can recognize the individual. Thank you very much.